Today will be our first session on neuroimaging technologies. In this session, we will start by looking at how we navigate our way around the brain. The focus is primarily on the different neuroimaging techniques used to examine brain function. Specifically, we will look at single unit recording, electroencephalography, magnetoencephalography, positron emission tomography, and transcranial magnetic stimulation. We will not look at structural and functional magnetic resonance imaging here because we cover it in more detail in one of our later sessions. There are different systems and labels used for navigating our way around the brain, each of which has its own merits. We will look at anatomical labels, functional labels, and Brodmann areas used to navigate our way around the cerebral cortex, and we will also look at white matter tracts. The brain is extremely complicated. We know by now that it consists of about 50 billion neurons, each of which can be connected with up to 10,000 other neurons. The amount of synapses in the brain therefore reaches the trillions. The brain itself we can think of as being composed of two types of tissue, grey matter and white matter. The grey matter consists of the cell bodies, which are the neurons, and the white matter contains the myelinated axons that connect with other neurons in the brain. The brain is highly convoluted, with ridges which we call gyri and fissures which we call sulci. The brain also has two cerebral hemispheres, a left and a right hemisphere, and the brain can be divided into four lobes, the frontal, temporal, parietal and occipital lobes. And within each lobe there are various anatomical labels that correspond with certain regions. These labels are based on the sulci and gyri in the brain. For instance, you may have heard terms such as temporal poles, as well as the fusiform gyrus, hypothalamus, hippocampus, and inferior frontal gyrus. The sulci are also labelled, for example the central sulcus and lateral sulcus. They are all used to help us navigate our way around the brain. The sulci and gyri are also used when trying to match people's brain anatomy with one another during neuroimaging approaches, so that we can compare activation in certain cortical regions across subjects. And we'll be going through this in more detail. Regions might also be based on labels of function. For instance, because the occipital lobe is almost entirely dedicated to visual processing, the occipital lobe is sometimes referred to as the visual cortex. Also, we refer to motor cortex and sensory motor cortex on the basis of functioning of that region of the brain. Also, there is Broca's area, named after the French surgeon for discovering that a particular region of the brain is involved in language. There are also systems in the brain that refer to a number of cortical regions, such as the limbic system, which is made up of regions such as the amygdala, hippocampus, limbic cortex, thalamic nuclei, and other regions lying either side of the thalamus. It appears to be primarily involved in emotion and the formation of memories. And even some of these regions can be considered as belonging to other structures. For instance, the hippocampal formation is thought to comprise the hippocampus proper, dentate gyrus, and subiculum. So we can see that brain regions can also belong to one or more brain systems, which may be defined on the basis of the function that it performs, such as we've seen with the limbic system. We might add to this complicated scenario by thinking of the ways in which cells are laid within the cortex. The grey matter contains the cell bodies of the neurons as well as glial cells. And the cortex is made up of up to 6 layers of cells. Each layer differs from another in terms of the size and density of the cell bodies of pyramidal pyramidal cells, which are pyramid shapes, and stellate cells, star-shaped, and the relative proportion of pyramidal and stellate cell bodies. The German neurologist Brodmann, who was around during a similar period to Broca and Wernicke, produced a cytoarchitectonic map of the brain based on the relative distribution of cell types across cortical layers. Approximately 52 areas labelled BA1 to BA52 are defined. Areas are labelled in a circular spiral starting from the middle, and this means the middle of the brain, which we can see here. Over the years, the map has been modified, but Brodmann has made one of the most successful attempts to define regions of the cerebral cortex. And to add to this, there are now also names given to different networks of white matter tracts. The most commonly known one is the corpus callosum, which is the bundle of white matter fibres connecting the two cerebral hemispheres. But there's also the arcuate fasciculus, which we can see here in red, connecting Broca's area with Wernicke's area. Developments in neuroimaging technologies, primarily diffusion tensor imaging, performed using MRI scanners, can be used to explore white matter tracts in the brain. And we will cover this in more detail in our session on magnetic resonance imaging. When we look at an individual brain, we can see the anatomical landmarks. This requires manual identification of our region of interest in each individual brain. For instance, when we want to locate Broca's area, or the pars opercularis and pars triangularis making up Broca's area, we can do this based on the sulci and gyri in the brain. But this is very costly in terms of time, and sometimes regions of interest may cross functional boundaries. So most researchers use an alternative approach based on transformation to a particular brain atlas coordinate system. So to perform group studies using functional imaging data, the individual brain images are usually transformed into a common coordinate space. The most widely used spaces in the neuroscience community are the Talairach space and the Montreal Neurological Institute space, which is MNI space. The Talairach coordinate system has become the standard reference for reporting the brain locations in scientific publication, even when the data have been spatially transformed into different brain templates, for example MNI space. When expressed in terms of individual subjects, the mapping of a coordinate in MNI space to the Talairach space generates distinct coordinates for different subjects. The Talairach space is based on a stereotaxic atlas of the human brain published by Talairach and Tourneau. The major criticism about the Talairach atlas is that it was created based on the post-mortem brain of a single subject, which is not a good representation of the neuroanatomy for the general population. To allow better representation of average neuroanatomy, the MNI created an average brain template based on the MRI scans from several hundred individuals. Since the Talairach coordinate system has become the standard reference for reporting the brain locations in scientific publication, researchers often need to report their findings using this system, even though their data were analysed in different coordinate space, such as the ICBM 152 template, which is the International Consortium of Brain Mapping Template, which is in MNI space. The transformation of MNI coordinates to Talairach space is no easy problem, and Shao and McIntosh explain in some detail how to go about doing this. Neuroimaging is becoming increasingly popular. This graph, taken from the Wellcome Trust, shows the number of papers published using neuroimaging technologies between 1989 and 2008 as a percentage of the total number of papers published. And we can see, in 2008, neuroimaging research accounted for approximately 1% of all total research in the world. So we can see that neuroimaging research is becoming increasingly popular. And this graph, also taken from the Wellcome Trust, shows the increasing trends for publishing neuroimaging research between 1990 and 2008 in the top 10 countries. We can see that Germany, who have long been interested in the physiological response, are in the lead when it comes to neuroimaging papers, but the UK is not far behind. We'll have a look at CAT scanning now. We include CAT scanning here for completion, so that you can really understand where the signal is coming from with CT scanning, what it's actually doing, and why we might not use this approach to look at brain functioning. So CAT scanning, sometimes referred to as CT scanning, provides a form of imaging known as cross-sectional imaging. The origin of the word tomography is from the Greek word tomos, meaning slice or section, and graph, meaning drawing. A CT imaging system produces cross-sectional images or slices of anatomy, like the slices in a loaf of bread. The cross-sectional images, which we can see here, are used for a variety of diagnostic and therapeutic purposes. So how does a CT scanner work? The patient is first placed on a motorised table, which moves through a circular opening in the CT imaging system. As the patient passes through the CT imaging system, a source of X-rays rotates around the inside of the circular opening. A single rotation takes about 1 second. The X-ray source produces a narrow fan-shaped beam of X-rays used to irradiate a section of the patient's body, which we can see in the figure here. The thickness of the fan beam may be as small as 1 millimetre or as large as 10 millimetres. In typical examinations, there are several phases, each made up of 10 to 50 rotations of the X-ray tube around the patient, in coordination with the table moving through the circular opening. The patient may receive an injection of a contrast material to facilitate visualization of vascular structure. Detectors on the exit side of the patient record the X-rays exiting the section of the patient's body being radiated as an X-ray snapshot at one position or angle of the source of X-rays. Many different snapshots or angles are collected during one complete rotation. The data are sent to a computer to reconstruct all of the individual snapshots into a cross-sectional image or slice of the internal organs and tissues for each complete rotation of the source of X-rays. The gradient of shade produced by the X-rays in a CAT scanner indicate the different densities of tissues and fluids. Bone and blood appear white, while CSF appears black, and neural tissues, in other words brain cells or neurons, lay somewhere in between, so they are a shade of grey. Because the CSF comes up black, dark areas can indicate edema and infarction, whereas bright areas indicate calcifications, hemorrhage, and bone trauma. And we can see here an example of how CAT scanning might be used to help us understand what is happening physically in the brain in different patient groups. Here are two images. One is of the brain of a patient with Alzheimer's disease, and the other is of a normally aged brain. There are differences in grey matter between the two brains, with the Alzheimer's brain, which we can see here on the left, showing a substantial loss in grey matter. The cortex around the brain appears darker because there is more CSF here as a result of the loss of grey matter. We can see that it's much darker than the grey matter around the healthy aged brain.
A CAT scan is used to detect both bone and soft tissue damage. During the procedure, three-dimensional anatomical imagery is created by utilizing X-ray technology. X-rays are used to create images by shining a high-energy electromagnetic beam through the patient's body. This beam casts a shadow onto an X-ray film. A photograph of the internal structures is created through the shadows cast by the varying tissue densities. Softer tissue allows more X-ray light to pass through, while harder tissues, such as bone, allow less light to pass through. While X-rays are limited to two-dimensional renderings, CAT scans are able to generate 3D views by rotating a conventional X-ray beam in a spiral around the patient's body. This series of rotations around the body creates image slices. A computer gathers, compiles, and translates these images into 360-degree view of a targeted area, or even the entire body.
While CAT scanners are useful for helping us to visualize brain structure and other types of tissue and bone, they do not allow us to investigate neural functioning in the brain. They cannot show us what is happening in the brain as a result of a stimulus, event, or task. To understand more about neural functioning and how the brain works, we need to use cognitive neuroimaging techniques. So let's turn our attention to those now. You've probably come across all of these different methods at some point. It can be quite confusing for a lot of people because they think each one is looking at the brain in the same way, but they are not all looking at the same thing. So which of these techniques is best for studying the brain? Well, there's no simple answer to that question. They all have their advantages and disadvantages, and they are all collecting different sorts of measurements. The best technique really depends upon our research question. You may have noticed that single cell or single unit recording isn't up there. I will briefly introduce this method to you, but given its invasiveness, it is confined to animal studies and so is not routinely used to look at brain functioning in psychological research. So we can see that there's different methods in cognitive neuroscience and different methods to look at brain functioning. So far, we have used the terms invasive and stimulation, but these are not the same thing. We can see here that the only invasive methods used are single-cell recording and PET, but neither of those involve stimulation of the brain. The only method that does involve stimulation is TMS, and this is not invasive. While single-cell recording is reserved for use on animals, PET is used on humans, though typically patient populations rather than healthy participants. EEG and single-cell methods measure the electrical activity that results from neural activity. PET, fMRI and NIRS are based on the hemodynamic response, which is based on the premise that an increase in neural activity results in an increase in blood flow to the area of neurons in the brain that are actively engaged in the task in question. And the methods also differ in terms of their temporal resolution. So let's move on to single unit recording. Single unit recording permits the study of a single neuron. By measuring changes in the responsiveness of a neuron to changes in a stimulus or changes in a task, it is possible to make inferences about the building blocks of cognitive processes. What is being measured in single unit recordings is the action potential of a single cell. A very small electrode is implanted into either the axon itself, referred to as intracellular recording, or the outside of the membrane, which is referred to as extracellular recording. The technique would involve counting the number of times that an action potential is produced, so spikes per second, in response to a given stimulus, such as a face. One of the best known applications of this technique was by Hubble and Wiesel, who did research on the neurophysiology of basic visual processes in cats and monkeys. They were eventually awarded the Nobel Prize in Medicine in 1981 for this research. So let's think. Neurons need to be able to represent how light or dark something is, as well as colour and edges for perceiving shape, changes in brightness and colour to represent movement and depth. The neurons in the primary visual cortex, area V1, transform the information in the LGN, the lateral geniculate nucleus, into a basic code that enables all of these types of visual information to be extracted by later stages of processing. And as with many great discoveries, there was an element of chance here. Hubble and Wiesel noted that an oriented crack in a projector slide drove a single cell in V1 wild. In other words, it produced many action potentials. They then systematically went on to show that many of these cells responded only to particular orientations. Now, single unit recording is clearly an invasive method. It is impossible to measure the action potential of a single neuron from the scalp because the signal coming from a single neuron is too weak and the signal coming from the many other thousands of neurons surrounding that neuron is too high. It would also be too difficult to localize from the scalp, as the spatial localization from the scalp would be poor, as is the case with EEG, for example, which we'll come to look at next. The single-cell recording technique is a much more fine-grained technique than other neuroimaging techniques. One advantage is that using this approach, neuronal activity can be obtained over time periods ranging from small fractions of a second up to several hours or even days. However, the technique can only provide information about activity at the level of single neurons, and so other techniques are needed to assess the functioning of larger cortical networks. Because of how invasive single-cell recording is, it is confined to animal studies. So let's have a look at what EEG does. EEG stands for Electroencephalography. In EEG, electrodes are attached to the scalp. EEG detects the electrical activity of neurons from the scalp, and the signal obtained comes from a population of neurons, as the signal coming from one neuron is too weak to be detected from the scalp. Also, The signal coming from a population of neurons gets weaker the further away it is from the source. This makes the method better for measuring neuronal activity in the outer cortex as opposed to subcortical structures. Each channel of EEG activity is recorded from disc-shaped electrodes which are taped to the scalp. The number of electrodes used varies. A 19 to 32 scalp EEG system is not a reliable localization method, whereas a 128 to 256 scalp EEG system is considered to be much more sensitive and reliable at localization. To obtain an EEG measure, you need to compare the voltage between two or more different sites. A reference site is often chosen that is likely to be relatively uninfluenced by the variable under investigation. One common reference point is the mastoid bone behind the ears, or a nasal reference. Another alternative is to reference to the average of all electrodes. The electrodes themselves are arranged at various locations on the scalp, and often described with reference to the so-called 10-20 system of Jasper 1958. The 1958 10-20 system is an international system that was designed to allow standardized reproducibility across studies, basically to be able to identify that the point being examined is the same across all subjects. The electrodes are labelled according to their location and the hemispheres involved. So in terms of location, we use the labels F, P, O, T and C. So F stands for frontal, P for parietal, O for occipital, T for temporal and C for central. And in terms of hemispheres, odd numbers represent the left hemisphere and even numbers represent the right hemisphere, and Z is used for the midline. For example, the O2 electrode is located over the right occipital lobe because we have O for occipital and 2, which is an even number and is localised to the right hemisphere. And if we said the FZ electrode, that would be located in the frontal lobe in the midline. So this would be located over the midline of the frontal lobes. The activity recorded at each location cannot necessarily be attributed to neural activity near to that region. Electrical activity in one location can be detected at distinct locations. Because EEG signals decrease in amplitude as they spread from their source, a comparison of signals recorded from various sites on the scalp can sometimes indicate the origin of particular waves. This is why it is usual to record EEG activity from many sites simultaneously. EEG provides detailed information about the time course of brain activity. It provides a continuous measure, and it can show brain activity in real time, so as it's actually happening. The typical situation is that we want to know what is happening in the brain in response to a particular stimulus. So we might present a stimulus to our participant and see what change in electrical signal is being produced in the brain. Sometimes the electrical signal can be messy or noisy. This is because it is difficult to distinguish between the normal brain's activity and the response that accompanies the perception of a stimulus. If we were to present a sound, for example, to our participant, the signal produced by that sound might be so small that it is difficult to detect from all the background noise that is going on in the brain. We would probably need to present the signal more than once, or the stimulus or tone more than once. We average the signals across trials to produce one clear signal or revoked potential, often referred to as an event-related potential. The potentials are event-related because they are related to a specific event such as a movement or visual stimulus or sound. The point of averaging across many trials is to produce a clearer signal. And we can see in this graph that when we average the signal for background noise across trials, what we are left with is a relatively flat signal. In other words, no change in signal, which we can see here. So these are all the times that the background noise is being collected, and when it's averaged across many different times, we get this average background EEG signal. Whereas when we average our signal related to our task, which we can see here, we end up with a nice average evoked potential where the signal is clear, which we can see here. So signal to noise ratio is low in a single trial, and each of these represents a single trial, but it is much higher when the signal is averaged over many trials. So this means that when different EEG waves are averaged relative to presentation of a stimulus, for example a tone, then the signal to noise ratio is enhanced and an ERP is observed. We can see here in this graph the average EEG signal to 1 trial, 10 trials, 50 trials and 100 trials. We can see that the signal is much clearer when 100 trials are performed. The graph consists of a series of positive and negative peaks. This is done for each electrode, so we get an ERP profile for each electrode. The positive and negative peaks are labelled P and N and are accompanied by either the corresponding number in which they appear, for example P1, P2 and P3, referring to the first, second and third peaks, or they can be labelled with P and N and the approximate timing of the peak, for example P300 and N400, and these refer to a positive peak at 300 milliseconds and a negative peak at 400 milliseconds. Whether the peak is positive or negative, in other words its polarity, is of no real significance either cognitively or neuropsychologically. What is of interest in ERP data is the timing and amplitude of the peaks. We can see that EEG activity when we monitor continuous EEG appears as a wave. Certain waveforms are associated with particular types of consciousness or particular types of cerebral pathology, such as epilepsy. For example, alpha waves are regular 8 to 12 per second high amplitude waves that are associated with relaxed wakefulness. The frequency of beta waves ranges from 12 Hz to about 30 Hz, in other words, 12 to 30 waves per second, and are closely linked to motor behaviour. In other words, they are generally attenuated during active movements. Low amplitude beta, with multiple and varying frequencies, is often associated with active, busy or anxious thinking and active concentrations. And we can also convert this EEG wave information into a brain map. Sometimes what researchers do, because of the poor spatial resolution of EEG, is they collect a structural MRI scan, which we will be talking about in another lecture, which is a high-resolution structural scan of the brain. And the researchers will then apply this EEG signal onto the structural MRI scan. This allows the different electrodes to be matched up to the correct points on the brain, and the signal obtained from each electrode would be imposed on the brain, which we can see here. This image gives an illustration of the different steps involved in converting the EEG and MRI scans into a map of activity. So we can see EEG data here and MRI data here. The high-resolution EEG data is first collected, and spikes are manually selected from the EEG data. A 256-channel system was used here. The spikes are then averaged, and the signals are then mapped onto the scalp. On the other side, we can see what is happening with the MRI images. So the MRI data is collected. The images are then segmented into different types of neural tissue, so into grey matter, white matter, and cerebrospinal fluid. And it's the grey matter segment that's used. The activity from the EEG is then mapped onto the grey matter segment using a type of inverse solution calculation. More information about this workflow can be found in Brodbeck et al. 2011. EEG has many different applications. In clinical settings, EEG is most often used to diagnose epilepsy. Brodbeck et al. 2011 have shown that a 128 to 256 channel EEG is considered to be a highly reliable tool for pre-surgical epilepsy evaluation. Its main strengths are its high temporal resolution, including its ability to monitor neural activity in real time, and it is non-invasive and it is cheap compared to other brain imaging technologies such as MRI and MEG. However, it has poor spatial resolution, and the signal is influenced by cell layering. For example, pyramidal cells are well aligned and fire together. So let's move on to MEG, which stands for Magnetoencephalography. MEG is used to study brain function and measures the magnetic field produced by the electrical activity in the brain. The size of the magnetic field by the brain is relatively small compared to the Earth's magnetic field. MEG is also based on the signal generated by a population of neurons rather than a single neuron. MEG provides an accurate measurement of brain activity because the skull is virtually transparent to magnetic fields. Thus, magnetic fields are little distorted by intervening tissue, which is an advantage over the electrical activity assessed by EEG. MEG has excellent temporal resolution in the region of milliseconds, and its spatial resolution is similar to EEG, which is not as good as MRI. Similar to EEG, an MRI is required to map activity onto. And MEG is very expensive. It includes using a superconducting quantum interference device, or SQUIDs, which use liquid helium. It also requires Faraday caging to shield any outside magnetic interference. We can see here in the image on the left areas of activity which are picked up by the SQUIDs, or superconducting quantum interference devices, during a somatosensory task. The waves represent the signal collected by each sensor on the MEG machine, which we can see circled here. These are the significant areas. And the idea is then to convert this into an image map. The image on the right shows activity, this time for a somatomotor task. The signal collected by MEG is imposed on an inflated map of the brain.
So there's this thing in your head called a brain, and it seems to be where thoughts and feelings and memories and perceptions and everything else has happened. So it would be great to know how it works. The obvious way to see how something works is to pull it out, open it up, and have a look, but when you do that with a brain, it stops working and tends to upset its owner. So what we really need are ways to study brains while they are working inside people's heads, which isn't easy, but can be done with a range of different brain imaging techniques. One of the popular techniques is MEG, Magnetoencephalography, which is much less scary than it sounds. Magneto means magnetism, encephalo means brain, and ography means images, so magnetoencephalography means images of the magnetism of the brain. Because the brain is made up of billions of cells called neurons. When the brain does stuff, these neurons are sending electrical signals to each other, which, like all electrical signals, generate magnetic fields. Magnetoencephalography measures and locates these magnetic fields, which indicate the electrical activity of neurons, which is the activity of the brain. With magnetoencephalography, we can see the brain working as it is working inside someone's head. The magnetic fields caused by brain activity are tiny and faint, so we measure them with highly sensitive magnetism detectors called squids, just like you'd use a highly sensitive microphone to measure a very quiet sound. If you were measuring a very quiet sound, you'd have to do it in a very quiet room, or else you couldn't hear the sound over the noise of air conditioning next door and cars down the road. And you get the same problem detecting the brain's magnetism, of the magnetism coming from nearby phones and computers and from the Earth itself. To block out this noise, imaging is performed in a shielded room built with thick layers of metal that block magnetic fields from outside. Another complication is that squids, our supersensitive magnetism detectors, only work when they're cooled down to about minus 270 degrees Celsius, which is almost the absolute coldest that it is possible for anything to be. Liquid helium can be used to keep the squids this cold. The squids are then arranged in a sort of a helmet, with a few hundred of the detectors held right up close to the head. Some specialized systems have smaller helmets to better fit children. The squids do need to be close because they only detect brain activity a few centimeters into the brain, and even then only some kinds of brain activity. But overall, MEG is still a pretty great brain imaging technique, partly because the process is so easy. The subject just sticks their head in the helmet, lies very still, and a detailed record is made of their brain activity. It's non-invasive, meaning we don't have to cut anyone open or inject anything or expose anyone to radiation, which a lot of the other techniques do involve. It also has really good spatial and temporal resolution, meaning it captures a lot of detail and can do so thousands of times per second, so we can see changes in brain activity moment by moment. Techniques like EEG have less detail, and MRI can only take an image every second or two. It's important to keep in mind, though, that MEG doesn't show the physical structure of the brain, just its activity. So MEG scans are often combined with MRI scans so we can see both together. But yeah, MEG has a lot of advantages as a brain imaging technique, and because of these advantages, it's widely used in research and diagnosis, where it can show which bits of the brain are active when people do different things, and which parts of the brain aren't working in patients. Some of the really cool things you can do with MEG include brain-computer interfaces, where a computer can monitor and respond to brain activity. In one study, a computer was set up to move the cursor on a screen when a particular kind of brain activity was detected by MEG. Stroke victims who couldn't move their hands learned to produce that kind of brain activity. They were able to move the cursor with their minds. When the computer was connected to a mechanical hand, the stroke victim could open and close the hand with their brain activity. Similar stuff has been done with monkeys controlling robotic arms. With this kind of technology, we can potentially restore movement to people who can't move and communication to people who can't speak. We can even work out what people are seeing and thinking by measuring their brain activity. Much of this research involves implants in the brain rather than MEG, but it's great when MEG can be used because it's so easy and non-invasive. But yeah, the point is that technologies like MEG are taking us closer and closer to Luke Skywalker robot hands and mind reading. In summary, magnetoencephalography works by using supersensitive magnetism detectors called squids to measure and locate the electrical activity of the brain. You've got to do it in a shielded room and with special cooling, but it's relatively easy to do and has great spatial and temporal resolution. Thanks for watching. Subscribe for more quick, clear explanations and analysis on stuff like Game of Thrones. Cheers.
We'll now move on to positron emission tomography, or PET. Positron emission tomography is based on the detection of positrons, which are the anatomic particles emitted by some radioactive substances. In a PET scan, radioactive tracers are used. This involves attaching a radioactive atom to chemical substances that are used naturally in the body by the particular organ or tissue in question during its metabolic process. For example, in PET scans of the brain, a radioactive atom is applied to glucose, which is blood sugar, to create a radionuclide called fluorodeoxyglucose, or FDG. Because the brain uses glucose for its metabolism, FDG is widely used in PET scanning. The radioactive tracer is injected into the body, which then rapidly gathers in the brain's blood vessels. When part of the cortex becomes active, the tracer moves rapidly to that place as the active neurons use up the glucose. The PET scanner slowly moves around the part of the body being examined. Positrons are emitted by the breakdown of the nuclear, the radionuclide. Gamma rays are created during the emission of positrons. The scanner then detects the gamma rays. A computer analyzes the gamma rays and uses the information to create an image map of the organ or tissue being studied. The amount of radionuclide collected in the tissue affects how brightly the tissue appears on the image and indicates the level of organ or tissue function. If a PET scan is taken of a patient who engages in reading for about 30 seconds after the injection of the radioactive substance, the resulting scan will indicate the areas of the brain that were most active during the 30 seconds of activity. The amount of radioactivity involved in PET is very small. The tracer has a half-life of only 2 minutes, although it takes 10 minutes for the tracer to decay almost completely. PET has reasonable spatial resolution. It can locate an active area of the brain within 5 to 10 millimeters. But PET suffers from some major limitations. It has poor temporal resolution. PET can only indicate activity over a period of between 30 to 60 seconds. It cannot assess rapid changes in brain activity associated with most cognitive processes. PET provides only indirect measures of neural activity. Changes in regional cerebral blood flow are thought to reflect changes in the spatial distribution of the positron-emitted radioisotopes, and this might be more applicable to the earlier stages of processing. And PET is an invasive technique because participants are injected with radioactively labelled water, which makes it unacceptable to some potential participants. Let's have a look now at a video that shows PET in practice.
PET, or positron emission tomography, captures images of minuscule changes in the body's metabolism caused by the growth of abnormal cells. Arrive 15 minutes prior to your exam.
Hi, Mrs. Craven.
Patients undergoing a PET scan will have restrictions on what they can eat or drink before the procedure. Contact the staff for detailed information on diet, especially if you have diabetes. The technologist will ask you to change into a gown or hospital scrubs. Do not bring children or pregnant women with you due to the radioactive material that will be injected. The technologist starts an intravenous line and checks your blood sugar. Then the radioactive tracer, called FDG, is injected. It will allow the radiologist to see images of how your cells take up the radio tracer. The radio tracer circulates in the blood for about 45 minutes, and then you'll be taken to the PET scan area. During the PET scan, you should relax and stay still. Most scans take about 45 minutes. You may listen to music during the exam.
You can wear your headphones. Keep your eyes closed there. We're just going to take a little test picture, so I need to put my laser lights on.
If a muscle relaxant is planned for your exam, be sure you have someone to drive you home. The PET scan can be used in conjunction with CT, or computed tomography images, that allow physicians to pinpoint the exact location, size, and shape of the diseased tissue or tumor. PET scans are used most often to detect cancer and to show whether cancer treatments are working. PET scans of the brain are used to evaluate patients who have memory loss, seizure problems, or brain tumors. PET scans of the heart can be used to check blood flow to the heart muscle. and help evaluate signs of heart disease. After the exam, you will not feel anything from having the radio tracer in your body. Drink plenty of fluids to help flush the radio tracer. A radiologist will review and interpret the findings and send a report to your physician, who will then give you the results.
Now let's move on to transcranial magnetic stimulation. TMS is a method of inducing a change in neuronal activity. It is based on the principles of electromagnetic induction of an electric field in the brain. The field can be of sufficient magnitude and density to depolarize neurons. When TMS pulses are applied repetitively, they can modulate cortical excitability, decreasing or increasing it depending on the parameters of stimulation. In practice, TMS involves positioning a magnetic coil next to the skull. Magnetic stimulation then temporarily turns off part of the brain while the effects of the disruption on cognition and behavior are assessed. TMS is one of the only methods that we have of establishing causation rather than simply observing correlation. And we have a video of how TMS is being used in practice.
If we start at the back of the brain, we've switched off my visual cortex, so made me unable to see things. If we move a little bit forward in the brain, we've interfered with people's ability and my ability to see faces. A little bit further forward, we've interfered with the ability to perform actions. Start moving your finger. Quite odd. It's not your finger anymore.
It's not my finger anymore.
We've interfered with the ability to compute with numbers.
6, 7, 8.
Okay.
Ooh, yeah. Yes.
We've interfered with the ability to speak.
London Bridge, London Bridge, London Bridge is falling down.
You think of something you can do, and we've basically interfered with it with this machine.
2, 3, 4, 5, 6, 7, 8, 9.
So what's the problem?
I don't know.
There's quite a lot of extra reading to go with this session, and I would recommend having a read through most of these articles. I hope you've enjoyed this session.